Well, hello everyone. My name is Javier Hidalgo Jimenez. I hold a bachelor's degree in biomedicine, and I'm finishing right now my master's degree on neuroscience. I'm going to present here the molecule within, a tale of science's journey into mammalian DMT. I chose this topic because after a personal experience with DMT, I was very intrigued by the fact that many animals, including humans, could synthesize DMT, and I think it's important to bring this issue to discussion because actually I think it holds a lot of potential and it's rather stigmatized by scientific community. Well, DMT stands for dimethyltryptamine. It's a very remarkable psychedelic with intense effects, even at medium doses that are very short lasting. Within some minutes, the effects are disappeared because DMT is metabolized very quickly. And DMT is the simplest molecule the simplest psychedelic molecule there is. Actually, you don't need to be an artist to draw DMT's molecule, as you see. Perhaps because of this simplicity, it's a molecule that is ubiquitous in the living world. DMT is produced by many plant species like Psychotria viridis, for example, that is used in the Amazonian basin to cook ayahuasca. It's synthesized by DMT-related compounds. 5-MeO-DMT is synthesized by toads that are also very well known for their 5-MeO-DMT production. But also many mammalian species, including humans, are able to synthesize DMT from normally occurring compounds. And this ubiquity of DMT is not magic, it's not causal, and it's not a conspiracy of machine elves. It's biochemistry. I mean, DMT belongs to a family of molecules called monoamines, which in turn come from amino acids. Amino acids are in every single cell because they are the building blocks of proteins. Additionally, amino acids like tryptophan, for example, can be transformed into monoamines by removing the carboxyl group in their molecule. And like serotonin, for example, serotonin is a monoamine that is synthesized from the amino acid tryptophan. Serotonin and other monoamines have a very special function in animals, which is cell communication. And when they communicate neurons, we say they are neurotransmitters. Tryptophan can also synthesize DMT, or can also yield DMT, which in plants, for example, it's the precursor of auxin, that is a very relevant plant hormone. So once more, we've got this communication, this cellular communication role. But here, we're interested in if animals can produce DMT in a way that DMT is significant for the biology, for our biology. So the state of the art looks a little bit like this. There is two radical opinions and very little science being done on this topic. On the one extreme, we've got the most spiritual option that states that DMT is more or less the link between the soul and matter. In the other extreme, we've got the more skeptical option that states that DMT is not likely to have any relevant role for biology and physiology in mammals. How did we come? Well, my personal opinion is that DMT, indeed, yes, can have significant roles in our physiology, and I will try to explain why by going through the history of endogenous DMT research. Endogenous means that it's synthesized within, and in this case, is within our bodies. Okay, let's start from the beginning. In 1961, it was the first time that DMT, that mammalian tissue, which was the rabbit lung, was seen to be able to produce DMT from tryptophan. At that years, psychedelics were called psychotomimetics because they were believed to reproduce the symptoms of psychosis. And this is why many psychiatrists were interested in if maybe psychosis was due to an abnormal production of endopsychedelic compounds in patients with psychosis. So many studies analyzed blood, urine, and cerebrospinal fluid, which is CSF, That's a fluid that is in contact with the central nervous system, so it's believed to mirror the chemical composition of our nervous system. And they looked for DMT in all these three fluids, and they found that actually normal people, healthy people without any diagnostic of psychosis, had more chances to be positive for DMT in their samples than people with a psychosis diagnostic. So this meant that DMT the presence of DMT did not predict the fact of having psychosis or not, at least baseline levels. Here we have to note, because we will come to this fact later, that the DMT levels in the CSF were comparable to those of serotonin. This means that we would expect to find inside the nervous system as much DMT as serotonin. Coming back to the issue of psychosis, okay, we can't run out the possibility that DMT may underlie acute psychotic episodes. We know that acute stress rise DMT levels, and also we know that acute stress triggers psychotic episodes. So there could be a relation between psychosis and DMT through stress. Anyway, this stress-dependent DMT production inspired Dr. Rick Strassman. Strassman is a well-known doctor that drove very relevant research on human DMT pharmacology in the 90s, and he launched a book. He published a book called DMT: The Spirit Molecule, which has been probably one of the it's one of the two most influential works done in this research field, even though it's not a scientific publication. But however, in this book, Strassman hypothesized that DMT was produced in the pineal gland, and under life-threatening situations where that are for sure very, very, very stressful, DMT levels rise so much that they activate 5-HT2A receptors, which are responsible for psychoactive effects of all psychedelics. And this way, they would elicit altered states of consciousness. What DMT would elicit altered state of consciousness that were reported during near-death experiences. The pineal gland is actually a very, very small organ inside the brain, only a few millimeters length, which mainly function is the melatonin secretion. Probably the other most relevant work on the field to date is Nichols' paper on 2018, where David Nichols is a pope of psychedelic pharmacology, and he claims that the pineal gland is too small to produce psychoactive amounts of DMT. In a whole day, in 24 hours, the pineal gland is only produces around 30 micrograms of melatonin, and we need 25 milligrams of DMT to reach fully psychoactive effects. So this would mean that in a few hours or minutes before you die, your pineal gland must synthesize like around 10 times more DMT than the amount of melatonin it produces in a whole day, which is very, very unlikely. But David Nichols went further, and he argued that because DMT levels are only a trace amount, which is very, very tiny amounts in the brain, it is not likely that DMT have any relevant function in mammalian neurobiology, neurophysiology. Well, here we have to claim that actually CFS amounts of DMT as high as serotonin's amount, so we would expect to have considerable amounts of DMT in the CNS, and actually measuring properly DMT levels in a brain is very, very difficult, because every 6 minutes you have half the concentration you had originally of DMT, because it's degraded very quickly. And all these studies that Nichols cite are based on whole brain homogenates. Whole brain homogenates are like kind of whole brain milkshakes, where if you could have initially high local concentrations of DMT, let's say in the visual cortex or in the prefrontal cortex, and you dilute them by homogenizing the brain, well, these levels will be the final levels that you measure are much lower than the original. So it's technically very, very challenging to properly measure DMT levels. In 2019, there was, however, a very important publication that gave a lot of answers. In this essay, researchers attempted to measure DMT levels directly from the pineal gland with microdialysis, which is a technique that consists in inserting a tube inside targeting a brain structure, and you can, like this, you can collect liquid samples of the extracellular fluid and measure local concentrations of any chemical in real time. So you are measuring local concentrations and you don't let DMT to be degraded. And they found, supporting the previous findings in CSF, that DMT levels in the CNS, in the central nervous system, are as high or well, much in the range of those of serotonin. Further, they induced cardiac arrest to the rats to see what happened with DMT levels, and they showed that they saw that DMT levels rise. They were doubled, but serotonin levels were rise by 20-fold, by 20 times more than the original levels. Serotonin has much more, 10 times more affinity for 5-HT2A receptors than DMT. So if we want DMT to elicit near-death experiences, DMT must activate these receptors. But at equal conditions, serotonin is more likely to be in the serotonin receptor than DMT. We need 10 times more DMT, 10 times more concentrated DMT than serotonin to displace serotonin and activate the receptors. Here we are seeing that in near-death experiences, serotonin levels are 10 times higher than those for DMT. So it's very, very unlikely that under these situations, any DMT release could effectively activate 5-HT2 receptors to elicit near-death experiences. They went further and they removed the pineal gland of these rats by surgery, and they saw that DMT levels remained the same. So the levels they were measuring didn't come from the pineal gland. Where did the DMT came then? Well, DMT came from the structure they had to traverse to reach the pineal gland, which is V1, the primary visual cortex. This brain area is connected to the retina, and it's crucial for conscious visual perception. I find this very, very interesting because first, DMT and ayahuasca trips are highly visual. Second, V1 is rich in 5-HT2A receptors, so DMT is where 5-HT2A receptors are, which is very relevant for any putative biological function of DMT. And also EEG recordings and fMRI during visual stimulation, during normal awake visual stimulation of a normal person, is very similar to the recordings that we see during eyes closed DMT or ayahuasca trips. This suggests that maybe DMT could be kind of a neurotransmitter that would be involved, for example, in this case, in visual perception. However, as we've seen, we need high local concentrations if we want DMT to be active. The only way to reach these high local concentrations would be presynaptic release of neurotransmitters. If we want neurons to release DMT, then DMT must be stored inside synaptic vesicles, which are little packages of neurotransmitters that protect, concentrate, and release, enable the release of neurotransmitters. We don't know if DMT can be stored within synaptic vesicles, but in 1977, it was shown that DMT was present in subcellular fractions that were associated with synaptic vesicles. Subcellular fractionation is a technique that permits to separate different cellular components. Unfortunately, the actual content of these vesicle-associated fraction was not proved to contain vesicular synaptic vesicles. But however, in 2009, it was predicted by biochemical methods that DMT could be a substrate for VMAT2, vesicular monoamine transporter 2, which is a protein that takes neurotransmitters inside, in this case, monoamine neurotransmitters inside synaptic vesicles. So it's very likely that DMT is actually stored inside vesicles, which means that could be a neurotransmitter in the mammalian brain. And you may say, Javier, but we already have serotonin. What's the point of having two ligands for the same receptors? I would say, okay, yeah, serotonin and DMT both bind serotonin receptors. This is true. But while serotonin activates the overactivation of serotonin receptors by serotonin elicits a rather unpleasant and potentially deadly serotonin syndrome, overactivation of the same receptors by DMT elicit an ineffable altered state of consciousness. So why this happened is beyond the scope of this talk and would need another presentation just on this topic. But the point here is that serotonin actions and DMT's actions on the same receptors do not overlap. It wouldn't be the first time that drugs help us to understand our nervous system, our brain. For example, opioid drugs, because morphine mimics endorphins, led us to the discovery of the opioid system. The same thing happened with cannabis. The THC, which is very similar to anandamide, that is the endogenous ligand of cannabinoid receptors, led us to the discovery of the endocannabinoid system. Okay, so here it comes the hypothesis, which is a hypothesis. Well, we normally assume that psychedelics mimic serotonin, that the endogenous ligand that is mimicked by psychedelics is serotonin. But actually, if we take a look, psychedelics look like DMT, not like serotonin. Every psychedelic in the psilocybin, or in the tryptamine, sorry, and the ergoline classes contain the molecule of DMT within their within their molecular structure. So what if instead of being serotonin, it's DMT, the endogenous ligand that it's activated, that is mimicked by psychedelic drugs? This is not like, I mean, this would be not a proper system, like this endopsychedelic system wouldn't be a proper system like endocannabinoid system or opioid system with its own receptors, but rather it would be a subsystem inside the serotoninergic system that would be based on the particular activation of serotonin receptors by DMT-like compounds. This, by the way, wouldn't only revolutionize, well, the discovery of this would go much more beyond the realm of psychedelic research, and it would be very relevant for all neuroscience. Well, we're finishing now, summing up. DMT is widespread in nature, including the human body. Currently, we can't explain why we produce DMT. Baseline DMT levels do not predict psychosis. DMT is not produced in the pineal gland and is not likely to underlie near-death experiences. If DMT can be stored in synaptic vesicle, which is likely, it may be a neurotransmitter with roles such as visual perception. And DMT could be the ligand of an endopsychedelic system. I want to thank ICR's Foundation and my friend Jose Carlos Bouso, because they encouraged me in my inquiries on this topic. I also want to thank PsychedelX team for offering me the opportunity to share this talk with you. And thank you for your attention. This is my email. You can write to me if you want. And if you're interested in this issue and you want to go more in depth or to see the technical details, you can read my review on the topic. And that's it. Thank you very much. Goodbye.